Ruben, you want to make...
Hello.
I was
Hello, hi
going to say,
guys.
you want to make the introduction. Okay, hi guys. So we are continuing with our review of Life Science in a Nutshell notes. Um, so I believe this is the ninth part. So where we left off, we're going to continue. Me and Ruben here. Um, so we were looking at the interior of the cell. Okay, so the interior of the cell contains obviously a cell membrane and a cytoskeleton. So we were going over the parts of these things. Previously, we were learning about some important things about the membrane and its membrane proteins, and the proteins in the membrane are affiliated with um, the cytoskeleton of the cell, the interior. Um, so there's interactions between them, and this is how the cell changes its shape and moves around, as well as its organelles are able to be mobile within the cell during its lifetime. So we were learning about the parts of the cytoskeleton, which consist of mainly microtubules, um, microfilaments, and intermediate filaments. So, while in size-wise, microtubules are the thickest, then intermediate filaments and microfilaments, also known as actin filaments, are the thinnest in nanometers. So, nanometers actually is not 1 millionth, it's actually 1 billionth. I would like to correct myself. Um, so, nano, it means 1 billionth, so 10 to the negative 9. Millionth is micro, which is 10 to the negative 6. So, not to get confused. So, it's a very small unit, and these filaments are measured in those terms. So, for example, the intermediate filaments, which is like the in between, in the middle range, I believe the micro tubules were about 25 nanometers. So intermediate are about 11-ish. It says here 9 to 11 nanometers in diameter when fully assembled. So nanometers is a unit used in science, units of length, time, etc. Okay, so we're going to continue learning more about the cytoskeleton. This is a picture of F-actin, which is a microfilament. The subunit of microfilaments is actin. Ruben, you want to start reading that?
F-actin distribution in the cell cortex, as shown by rhodamine, phalloidin, staining of HeLa cells that constitutively express histone, H2B, GFP to mark chromosomes. F-actin is thus red, while histone H2B is displayed in green. The left-hand cell is in mitosis, as demonstrated by chromosome condensation, while the right-hand cell is in interphase, as determined by intact cell nucleus in a suspended state. In both cases, F-actin is enriched around the cell periphery. Scale bar 10 micrometers.
Beautiful. So this picture is very, very nice. It's bright, so we can see that how the actin and its shape is changing and becoming wider and expanding and becoming smaller and contracting in various stages of the cell cycle and cell replication, etc. So the cell cortex is another name for the what is part the cytoskeleton. It is defined as either the actin cortex or actomycin cortex. It's a specialized layer of the cytoplasmic protein on the inner surface of the plasma membrane of the cell periphery. So, so on the inside of the plasma membrane, on the periphery, you have the cell cortex functioning as a modulator of plasma membrane behavior and cell surface properties. Effect and distribution in the cell cortex is shown by rhodamine palladian staining of HeLa cells that constitutively express. So, so we just read that. Okay, so not to repeat what we said, but the cell cortex is also known as it is a layer of the protein, blah blah, blah blah. I don't know why. In most eukaryotic cells lacking cell wall, especially the cortex, is an actin-rich network consisting of actin filaments, myosin motors, and actin-binding proteins. The actomycin cortex is attached to the cell membrane via membrane anchoring proteins um, called DRM proteins and plays a central role in cell shape control. The protein constituents of the cortex undergo rapid turnover, making the cortex both mechanically rigid and highly plastic, so kind of flexible, two properties essential to its function. In most cases, the cortex is in a range of 100 to 1,000 nanometers thick. In some animal cells, the protein spectrum may be present in the cortex. Spectrum helps to create a network by cross-linked actin filaments. The proportions of spectrin and actin vary with cell shape. We're going to learn a bit more about spectrin in a bit. And the spectrin proteins and actin filaments are attached to transmembrane proteins by attachment proteins between them and transmembrane proteins. So the cell cortex is attached to the inner cytosolic face of the plasma membrane. In cells where spectrin proteins act in filaments form a mesh-like structure, much like a fishnet, except that it can be broken and reformed. In plant cells, cell cortex is reinforced by cortical microtubules as well underlying the plasma membrane. The direction of these cortical microtubules determines which way the cell elongates when it grows. So functions. Ruben, read this, please.
In mitosis, F actin and myosin to form a highly contractile and uniform cortex to drive mitotic cell rounding. The surface tensions produced by the actomyosin cortex activity generates intracellular hydrostatic pressure capable of displacing surrounding objects to facilitate rounding. In cytokinesis, the cell cortex plays a central role by producing a myosin-rich contractile ring to constrict the dividing cell into two daughter cells.
Cell cortex contractility is key for amoeboidal type cell migration, characteristics of unicellular cells, etc. So cytoskeleton is present in all cells. It's a complex network of interlinking filaments and tubules extending throughout cytoplasm from nucleus to plasma membrane. So we're going to continue in the next part. Bye.
Bye.